evidence for the future.
This man has acute myelogenous leukemia. He has flown from his home in Denver to the U.S. Public Health Service Hospital in Baltimore for new treatment with drugs.
But I was interested to know so that I could do some personal planning.
Yes.
Along these lines.
I think this is very important for you to take care of your own, make sure your house is in order.
Sure. I was wondering what the possibilities of a remission would be with the drugs which you have mentioned, and how long this remission would last.
With our present experience, we are getting remissions at somewhat over 1/3, 40% of patients. And it is true that when you do reach remission status, you are clinically and hematologically normal. There's no evidence of leukemia whatsoever. Now, our big problem is we are just stymied and are unable to maintain the remission too long. And exactly from case to case, how to predict how long it will last, I can't do. We do have some patients that have been in remission well over a year and a half, 2 years.
And this is a substantial time.
But there are many, many patients who are only in remission 1 or 2 months. But of course, let's say we do use donomycin, we do get a remission, and in the event that relapse does occur, we can use cytosine arabinoside. In fact, we have other drugs after that. One of the benefits of being here, at least, is that there are always new things on the horizon that are here, and if a breakthrough does occur, you have a good chance of participating in that breakthrough here.
We felt that
in medical research, there is always hope for a breakthrough. Can science find a drug to cure cancer? Cancer has been treated for some time. Many of the cancers which form solid tumors can be treated with surgery. If all the cancerous tissue is removed, the patient is cured.
Let's have another reciter.
The same is true of radiation treatments. Cure depends upon destruction of all the cancerous tissue. But sometimes the cancer spreads to other parts of the body. Leukemia, or cancer of the blood, is of course all over the body. In such cases, the ideal therapy would be a chemical which could spread through the body and kill every cancer cell. For more than a decade, there has been a national program to find and test anti-cancer drugs, administered and supported in great part by the National Cancer Institute at Bethesda, Maryland. The search is worldwide. There are 10 million natural products that might contain an anti-cancer compound. Near Nairobi, botanists gather leaves and the bark of trees to process and test against cancer. In the shallow waters of the Caribbean, marine biologists collect invertebrates like the West Indian starfish. As part of the nationwide cancer chemotherapy program, they process and test the starfish to find out whether it might contain drug materials effective against leukemias and solid tumors. The search goes on in North Carolina with the gathering of plants and grasses by botanists with the United States Department of Agriculture. Any of these could contain a valuable anti-cancer agent. To test these materials for anti-cancer activity, researchers might screen them in 1,000 animal tumors against 100 different human cancers. How can scientists determine what chemicals are in these dried twigs? How can such chemicals be tested safely in human beings with cancer? The unknown material sent in from the field is first processed and tested for anti-cancer capability in two so-called animal models: laboratory mice with a known leukemia and rats with a known tumor. These two types of cancer catch almost all actives. If the compound is an active, it will reduce the tumor mass, as compared with the tumor in a rat that got no compound. But if the compound kills tumor cells in a rat, it is likely to kill some normal cells too. To measure these adverse effects, researchers administer the compound to monkeys. After a suitable period, they remove a sample of the monkey's blood and subject it to a series of tests that reveal the effects of the unknown chemical on normal blood cells. For example, the coagulation time. Other tests show other effects on the blood and on the kidney, the liver and spleen, and particularly on the bone marrow, where red corpuscles and white blood cells are manufactured. Such tests indicate the chemical's toxicity and help define a safe dose for the test in human beings. If a compound shows anti-cancer activity, other researchers try to determine what it is composed of by means of column chromatography and many other tests. The chemical compounds flowing down this column, stratified in this way because of their unique molecular properties, may turn out to be potent anti-cancer agents. More likely, they will not. Only about one agent in 15,000 tested has proven effective. Chemists also attempt to synthesize new anti-cancer agents. This is a plastic model, enlarged thousands of times, of a molecule essential to the reproductive process in all cells. To produce one effective drug, researchers, over many months, removed one of the hydrogens and put a fluorine in its place, producing an antagonist that is absorbed by the cell as if it were the real thing, then destroys it. In the last test, a safe and effective dose for human beings is determined by preclinical pharmacologists. The moment of truth in the long search is the clinical trial. Will the leaves and grasses from Nairobi, or the compound synthesized in a laboratory, make a drug to control cancer in human beings? This is a regular staff conference in the Clinical Center at Bethesda. Case histories of patients on trial programs are presented. In this instance, the first patient on a new drug. Then the doctors discuss the implications of their findings for the nationwide research program. A doctor wonders where the first phase of drug study should be done.
In other words, maybe the phase 1 study should be done on patients with diseases in which there is no established accepted therapy that would benefit them.
Suppose you get phase 1 information, say in a solid tumor, and you get some idea of what the toxicity, but you really, is that going to apply in the patient, say, with acute leukemia or CML blastic crisis? And I think
well, at least you would have evaluated the drug under relatively clean conditions.
Yeah, but I think
It gets complicated, I think, when it involves the marrow, when the disease involves the marrow.
Much research is being done on the size of the dose. Large single doses kill cancer cells effectively, but they also kill some normal cells, so doctors must balance dosage against adverse side effects.
Chuck, we've had one patient who's been on 800, which is twice this dose. And what's happened to him?
He tolerated the single dose of 800 milligrams of meter squared quite well. He left the hospital. The second set of liver function tests are going to be done this week, but we haven't heard from him, and I venture he's doing quite well.
Combinations of 4 drugs are effective, but it has been difficult to follow up certain 4-drug programs with some of the new drugs, such as cytosine arabinoside.
Yeah, I think this brings up an important point about where phase 1 studies should be done, because we noted when we first started using cytosine arabinoside, it was after the 4-drug combination, after people relapsed, and we saw a significant number of patients with transient, but often quite severe hepatic damage with cytosine arabinoside. We were very concerned about it. Other institutions were starting right off with cytosine arabinoside in AML, and saw none of it. And I think this was probably from the same reason. The people that we had been in remission an average time of 9 or 10 months, and they got cytosine arabinoside, they were just more sensitive to the drug. Same thing applies with reinduction with a 4-drug combination. We saw a higher degree of hepatic dysfunction at reinduction with the same drug as we did in primary induction when the patient was first diagnosed.
Some cancers adversely affect the bone marrow, where red corpuscles and white blood cells are produced. By studying a sample of the bone marrow, doctors can follow the evolution of the disease and the effects of different drug programs.
What's the present bone marrow show, Bridget? I mean, did we get our own marrow, or are we relying on the marrow from Hodgkins?
Yes, we
And
have
cytogenetics?
No, we have not obtained a marrow yet.
He hasn't had one yet here, but he has our rods in the peripheral blood, so we're not in any doubt about the diagnosis.
What's the percent blast for only 5%?
To take a marrow sample, the doctor carefully anesthetizes the local area, then inserts the needle through the flesh into the end of the bone. And on into the marrow. A syringe withdraws a small sample of the marrow. This is placed on microscope slides, then examined. This marrow sample from a patient with acute leukemia is hypercellular, saturated with malignant cells. At a larger magnification, the malignant cells, called blasts, show an unusually large nucleus. After 2 courses of treatment with drugs, a marrow sample might show fewer blasts and increasing numbers of normal white cells, a sign of improvement. Later, in remission, when the disease is under control, there will be only 5% blasts. But the peripheral blood, the blood that is flowing through the body, is also a good indicator of the state of the patient's disease and the efficacy of treatment. A sample of the patient's blood is diluted and placed on a counter. The dials reveal the number of blood cells in a measured sample. A symptom of leukemia is an excessive number of abnormal white blood cells, ineffective against invading microorganisms. Cancer research needs many blood donors. Leukemia, for example, sometimes reduces the number of blood platelets, the tiny cells that maintain the integrity of blood vessel walls. Bleeding may result. But platelets can be replaced by transfusion. Whole blood is removed, centrifuged to bring down the heavier red cells, and placed on an extractor that squeezes the platelet-rich plasma off the top of the sample for transfusion into the patient. In this case, the son of the donor. Since both the drug and the cancer may affect the body's ability to resist invading microorganisms, many patients die of infection. For protection, some patients are placed in a plastic bubble that completely surrounds them at all times. Doctors conduct routine examinations even though they are completely cut off from the patient. Only filtered air and aseptic materials enter this sterile private world. Yet doctors can perform bone marrow and other operations. This laminar flow room, now under test at Bethesda, may someday replace the plastic bubble. It will repel microorganisms by continually blowing fresh filtered air across the patient. Doctors and visitors will enter and remain downstream. If the experimental drug cannot control the number of white blood cells, doctors use the continuous flow blood cell separator shown at the MD Anderson Hospital and Tumor Institute in Houston. The machine draws whole blood from one arm, pumps it through carefully sterilized tubes to a weight-sensitive buffer bag which controls the pump, and sends it to a centrifuge, which is similar to a cream separator. Because of the different densities of white cells, red cells, and other blood elements, they separate at different points along the radius of the disc. Unwanted cells are shunted aside and the rest returned to the patient. If a drug is successful against one kind of cancer in phase 1, it is tested against other kinds. Then it is sent for further tests to many hospitals around the country.
Was it irreversible? Does anyone know what the liver toxicity has been with the first imidazole, since that's been tried pretty extensively now in the Midwest?
There's not really been much mention made of liver toxicity in the patients that have been sent in that have been evaluated after therapy. So I would assume, although I don't know directly, because we haven't used the old drug here, that there really isn't a significant amount of liver toxicity with that particular kind.
Always there are the questions of scheduling, balancing dosage against toxicity.
I think that we ought to really be careful about how we evaluate the liver toxicity and the circumstances that occur, so we don't do this all over again.
Right. I agree with you very much on that. Now, the thing is, one of the reasons that we are happy with this type of schedule, that is giving one dose and then waiting to see recovery, is that it gives us time to back off and to let the toxicity abate. And I think it's probably the safest way to start out giving a drug.
And finally, the question of an adequate trial for each drug.
What would you consider an adequate trial for this drug? How much? How long?
What would you, in this particular patient?
Yeah.
Well, that's... Well, I think that I would think that in this particular patient, if the physicians in charge think it's due to liver, due to liver, then I suspect, liver toxicity is due to the drug, and you can say, well, we had to stop because of liver toxicity. But my only impression is that I'm not so sure that we can attribute it all to the dose, and would feel that the risk involved in giving them, say, a half dose would not be that great. It would still allow us to see if this drug is indeed going to be useful, and we're hoping that, that this drug will be useful in this particular individual.
Since July.
In clinical research, the doctor is concerned not just with a drug and an effective schedule of administration, but with a whole patient and his disease.
You given her donomycin how long?
She's gotten one dose.
One dose?
Exactly 1 week ago, and she's scheduled for her second dose today.
Have you ever had any trouble with your heart?
Not that I know of.
Never had a murmur as a child?
Not that I know of.
Have you found anything?
Her cardiac status is completely normal as far as we can tell.
But the doctor always returns to the key questions.
8,000 international units.
Which dosage has worked?
But this
Which
has varied
schedule
somewhat
has been most
to,
effective?
A level of 16,000 or 25,000, but in terms of concentration, it's not too different.
Well, she didn't respond at all to actinomycin D and methotrexate, or?
On one occasion, her titer became completely negative, that is 0, and subsequently came back up again, though. This was on actinomycin D. In the last few months, she's received 3 or 4 courses of methotrexate in infusion type, and her titers have gone down and come back up, but her general course has remained essentially unchanged. In the light of the natural course of this disease, it's fair to say that the methotrexate has been somewhat effective.
Did you ever decide in your mind whether she was really drug resistant or just perhaps a dosage?
I think it's difficult to say. If we could give the methotrexate more often, perhaps we might have a better effect.
What's been the limiting factor on
The limiting factor has been toxicity related primarily to ulcerations in your mouth, and which have been quite severe, although not complicated by infection so far.
She had ulcerations anywhere else with methotrexate?
No, not that we know of.
Got a rash?
She did have a rash on her skin, which I couldn't say was clearly related to methotrexate, but may have been. She has not had it with each dose. She's tolerated the therapy actually quite well.
Did you notice anything when you got your first dose of donomycin?
No, I was really surprised.
Pleasantly surprised?
Yes, very much so.
Well, it's going to be a long dosage schedule, and we've had some good luck with it, as Dr. Neidolf mentioned.
The guiding principle in all good clinical research is concern for the patient.
Jeff and I, we're not quite as relaxed as you are.
Well, I'm not relaxed.
How's the back doing today and the legs?
My legs hurts.
They do hurt.
Down here, yeah. My back don't bother me. I hadn't for a long time.
I wonder if you could roll over on your side and roll towards the window. We just want to feel your back. Let's see, it was a second
I want you to tell me, Mr. Simpson, if this, when and if this hurts you at all. Is that tender?
No, not yet.
That tender?
No.
That tender?
That's a little bit.
How about over here?
No.
Over here?
No.
Right here?
Well, I believe that's a little bit right there.
Here?
No.
Here?
No. A little bit right there. That's the end of your finger. Right there.
A lot or a little bit?
No, just a little.
See, I wonder if percussion would reproduce any of the pain down the leg. Let me see. Okay. Well, we finally got the results on the X-rays
Yeah, what
that
you got?
they did, and there does seem to be something that may well be causing the pain down the leg, but it's in this area.
Yeah.
We're going to talk with the radiotherapy people today and see if they can help us on it. We'll let you know later on today on that.
You let me know today, huh?
Yeah.
Today?
I think so. It'll depend when they'll be able to get to see you. Your next course of chemotherapy starts next week.
Next week?
Yeah. About Tuesday, Monday or Tuesday. Depending on what your blood count shows. It'll be, they think they can help you with X-ray combine the two.
Okay.
Okay. So maybe that's what's doing it.
You think so?
Yeah, I think.
You think you found
Pretty
it
good
back
chance,
here? Right in that sort of thing.
Yeah.
Well, everything else, you know, has improved.
Yeah.
Lump has gone down, the chest is better, and so forth. So if we can get this pain under control, I think everything should be a lot better in the system.
Okay.
So that's what we're shooting for.
All right.
Where has the National Cancer Institute program led? In advanced forms of Hodgkins disease, a 4-drug treatment produced complete remissions or temporary disappearance of all evidence of cancer in twice as many patients as ordinarily achieve remission with single drugs. A group of 35 children with acute leukemia were treated intermittently with massive doses of 4 drugs. The median survival of these children was 3 years, compared with a 3-month survival of leukemic children 20 years ago. The story of cancer drug research will continue next week in Part 2, The Battle in the Cell. Further information on cancer chemotherapy, write for a free booklet, Drugs versus Cancer, available from the Research Information Branch, National Cancer Institute, Bethesda, Maryland. These are normal human cells from prosthetic tissue living in a laboratory dish. They were filmed through a microscope that magnifies them 270 times. These are the same kind of human cells from prosthetic tissue, grown and filmed in the same way. But they are cancer cells. The cells, normal and cancer, differ in ways not yet fully understood. It is the task of the cancer chemotherapist, the scientist who hopes to control cancer through the use of drugs, to exploit those differences so that a drug can kill all of these cells without killing more than a few of these in the same human being. Cancer has been treated successfully for some time by surgery, in which the malignant tissue is removed, and by radiation, in which it is destroyed by cobalt and X-rays.
Did you drive yourself?
But sometimes cancer spreads to other parts of the body, even as the local area is being treated successfully. In such cases, new hope may be offered by chemotherapy, the use of drugs.
Well, what treatments have you had? I know you had surgery. Did you ever have X-ray therapy?
Mm-hmm.
When was that?
63, I think.
Right.
I couldn't say for sure.
And that was for a local recurrence of your disease, I think.
Yes.
And then did that hold it under control for a little while?
Yes, it did.
And then what happened?
Well, then it started all over again.
Have you ever received any other therapy? Very often patients with breast cancer get estrogen therapy, corticosteroids, cortisone.
The only thing I had was those pills.
And what's
what are those? Guys, I can't pronounce.
Diethylstilbestrol.
Yes,
Yes. These are pills. This is hormones that very often control cancer, breast cancer. Did this help?
Yes. When I was taking 2 or 3, it helped it better. And I had to stop them because I had started my periods.
Well, basically, the problem with your disease is that you did have localized disease, and you received surgery, and then for a local recurrence, radiotherapy. And I think now the problem is that the disease is just too widespread to be controlled by any of these local means. Where did Dr. Hunter tell you that you had disease?
My breast.
Eventually, what happens in cancer is that we do have to use drugs. And even after local therapy and after hormonal therapy. And you did have the hormonal therapy, and the disease has progressed to the point where your doctor did feel that you should go to a center that is more concerned and has more experience in drug treatment of malignancy. Right. Were you happy about going, or how did this strike you when you discussed this with him?
Well, kind of shocked me a little bit, but then I made up my mind I would. He advised me it was the best thing for me to do. He said if I could get in here, I would be very lucky. So I just told him I would.
For more than a decade, the National Cancer Institute has guided and supported a large national program of cancer chemotherapy. Scientists are studying the whole patient, the effects of many kinds of drugs on the course of his disease, and on the inner workings of cancer cells. A cell is visible to the human eye only through a microscope. Nevertheless, it is the building block of which all tissue is made. When new tissue is needed, the cell divides, each so-called mother producing 2 daughters, exactly like the mother. Each daughter produces 2 more daughters, also the same. When no more tissue is needed, the doubling stops, halted by an unknown biological signal. The cancer cell and all its daughter cells do not receive or do not obey the signal. They keep multiplying, far beyond what the body can tolerate. This relentless growth of non-functional cells can lead to illness and death. The experimental evidence of this is startling. Blood is withdrawn from the abdomen of a mouse with leukemia. It is diluted, and the cells are counted under a microscope. They are diluted and counted again until any given drop is likely to contain only 1 or 2 leukemic cells. With this instrument, called a micromanipulator, scientists at Southern Research Institute in Birmingham today can isolate a single living leukemic cell. Using a hair-like tube, the technician places several drops of the diluted material on a glass slide. The slide is turned over so the drops are hanging from the lower surface and is placed under a microscope. The technician locates a single drop. He carefully moves a pipette, a delicate glass tube, up under the slide. And with hydraulically operated controls that scale down his movements, he inserts it into the droplet. And withdraws the leukemic cell. The cell is transferred to a hypodermic syringe, a single cancer cell. It is injected into a normal, healthy mouse. Within 8 days, there is a noticeable abdominal swelling, evidence of the growth of leukemic cells. The cancer cells continue to increase, each mother producing 2 daughters, each daughter producing 2 more daughters, until after 10 days, the swelling in the abdomen is pronounced. By the 19th day, the single cancer cell will have proliferated to more than 1 billion cells, enough to kill the animal. But certain chemicals can stop this cancerous growth. In this laboratory culture, we see mouse leukemia cells dividing, with cleavage, as shown in the center of the screen, completed after the usual length of time. But when a drug is added, the reproductive cycle of the cells is violently disrupted. The cell in the center of the culture stays at metaphase, an intermediate stage in the division process, an unusually long time. Cleavage is irregular. Daughter cells are of unusual size and do not survive very long. Human cancer cells are also affected by drugs. These cancer cells in a laboratory dish are dividing in the usual way, as shown at far right. If an alkaloid from the periwinkle plant is added, cells proceed to the second phase of the cycle, as seen in the rounded cells, but cannot go beyond this phase. Mother cells cannot produce daughters, stopping the relentless reproduction that characterizes the growth of cancer cells. But it has been found that a given dose of a given anti-cancer drug destroys only a certain percentage of the cells in a living animal. Regardless of the absolute number of cancer cells, the percentage destroyed by the same size dose will always be the same. Suppose a given type of cancer cell doubles approximately every 12 hours. Once every 24 hours, we administer a drug which kills 60% of the cancer cells present. Again, the remaining cancer cells double. And again, we kill 60%. We have slowed them down, but they are increasing faster than we are killing them, and they will eventually increase in mass or number to the point where they kill the animal. We can increase the doses so that we are killing the cancer cells faster than they can multiply. But anti-cancer agents also affect normal cells. If the dose is too large, and if too many normal cells are killed while we are destroying the cancer cells, the animal may sicken and die. Is it possible to kill large numbers of cancer cells with drugs without killing too many normal cells? These normal mice are injected with an anti-cancer drug called cyclophosphamide. 24 hours later, the femur, or upper leg bone of the mouse, is removed. The ends clipped, and the bone marrow, the site of blood-forming stem cells, is washed out and diluted in a laboratory solution. To find out how the drug has affected the marrow and its ability to produce cells, the technician injects the diluted marrow into a mouse whose stem cells have been destroyed. 10 days later, after the injected marrow cells have had a chance to multiply, the animal's spleen is removed and fixed in buin solution. Marrow cells form nodules on the spleen. The number of nodules, in this case a dozen or so, indicates the number of cells surviving, a measure of the effects of the original anti-cancer agent on the normal cell. But what does the agent do to cancer cells? A lymphoma-bearing mouse is injected, and 24 hours later its bone marrow is removed. Again, following the same procedure used with the normal mouse, the marrow is diluted and injected into the tail of a mouse, which will serve as a kind of incubator or culture dish for the marrow cells. 8 days later, the spleen of the cancer mouse is removed. Again, the nodules are counted, an indication this time of the rate of survival of cancer cells after drug treatment. To get good data, 66 such experiments were conducted at the Ontario Cancer Institute, each one involving more than 100 mice. When the results were plotted, the curve showing the percent survival of normal cells as against increasing drug dosage looked like this. The curve indicating percent survival of cancer cells with increased drug dosage looked like this. There is no question that some drugs kill many more cancer cells than normal cells in the same living organism, and that kill ratios vary with dosage. So, dose amount, as well as timing, is important in the battle in the cell. But can we further improve this ratio, the ratio of cancer cell kill to normal cell kill? Just within the past couple of years, a new approach to this problem has been developed, based upon close observation of the life cycle of the cell. In addition to the phase during which the cell divides, there are several other cycle phases. Generally, cells appear to rest in one phase, synthesize genetic material in another, rest again, then divide. But timing is crucial. How can we find the length of each phase in the life cycle of a cancer cell? At Southern Research Institute, researchers use a radioactive material that is absorbed into cancer cells during the S phase. They inject it into laboratory mice with the solid tumor designated sarcoma 180. let the tumor grow, then remove it. In this tumor, how many cells have taken up the radioactive material? How many have passed through the S phase and on into mitosis, or cell division? After mincing the tumor, the technician suspends it in a measured amount of saline solution and carefully breaks down the tumor tissue in a laboratory homogenizer. Then the technician strains the mixture to remove large clumps of tissue. And places the liquid containing radioactive cancer cells into a centrifuge. Later, the packed cells are treated in other ways and are placed on glass slides, each carefully marked for future identification. To find out how many cells are radioactive and thus have passed through the S phase, the slide is converted into a kind of photographic plate. It is coated with emulsion and left in a dark room for 6 weeks. To count the cells that have absorbed the radioactive material, the researcher not only examines all portions of the slide under a microscope, but also studies it on closed-circuit television. Cells containing radioactive material are identified by the dark spots on the emulsion. On this slide, over 3,000 cells were studied and counted. From this experiment, the researchers determined that the life cycle of the sarcoma 180 cell is about 13.5 hours, and that the S phase lasts almost 8 hours. Important data in determining the best time to administer drugs that kill during the S phase. Of the thousands of drugs tested, some apparently kill only during the S phase of the life cycle. Yet their kill ratio is good. Of the thousands tested, some were found to kill throughout the life cycle, and they also can give good cancer cell to normal cell kill ratios. But when these different kinds of drugs are combined, they can give much better kill ratios. Much experimentation involves drug schedules. Experimenters at Southern Research Institute have developed ways to administer drugs steadily over long periods of time to mice with different kinds of cancer. A tube is inserted interperitoneally, and the mouse is allowed to run into his small chamber along with a dozen or so of his cousins. The experimental drug is drawn into a special syringe. and the syringe is mounted on a rack. One syringe for each mouse. Other syringes are filled with harmless saline solution for administration to a group of control mice. They will be watched to see whether the apparatus, rather than the drug, affects them in any way. Now each syringe is adjusted precisely to ensure administration of a carefully measured amount of drug. The mice in their stalls are given a final check. A timer is started, and the drug is fed through the tubes at a constant rate. In this apparatus, the drug can be made eventually to hit every cell in a cancer mass, no matter when or how long it is susceptible to the drug. But scientists thought it might be better to treat the animal with a single, stronger dose, followed by other doses at a later time, perhaps giving the normal cells more time to recover from drug damage before they are treated again. Not long ago, Southern Research Institute tested cytosine arabinoside in a batch of mice with leukemia. In the first experiment, they gave a measured dose of drug to hundreds of animals once each day for a period of 15 days. Some mice were used as controls. They got no drug. At the end of the experiment, the control mice died, as predicted on the basis of the growing knowledge of cell kinetics. The experimental mice, those that got the drug, lived much longer, although all eventually died. When the results were averaged out, it was determined that the daily dosage had increased the lifespan of the experimental mice substantially, although none were cured. But by simply changing the timing of the administration, the scientists got dramatically different results. They used the same kind of mice, mice which have been inbred for generations to enhance their similarity for laboratory purposes, and the same drug, cytosine arabinoside, and essentially the same size dose. But instead of administering the drug once a day, they administered it every 3 hours around the clock at 4-day intervals. Control mice again got no drug, and again they died according to estimates based on cell kinetics. The experimental mice, those that got the drug on the new schedule, continued to live happily. Some laboratory animals can be cured of cancer, but human beings are a far cry from laboratory animals. Can drugs be found or designed that will destroy the billions of cancer cells that sometimes spread through human beings? A computer program developed at the National Cancer Institute may help find the answer. Into the computer are fed assumptions about the generation time of a given cancer cell, the duration of the S phase, and the fractional cell kill of a given drug. In seconds, the computer can reveal, among other things, the number of cells remaining at the end of the dose period. With such information, drug programs can be tailored for individual patients. other progress is being made. The animal model for testing drug activity against cancer is giving good biological information that has a direct bearing on the disease in man. Although enormous problems remain, there is hope that continued research will lead to even greater life expectancy for even more patients. Information on cancer chemotherapy, write for a free booklet, Drugs versus Cancer, available from the Research Information Branch, National Cancer Institute, Bethesda, Maryland.